Welcome, everybody, to Cocktails and Fishtails. Y'all, this is the last presentation of 2024, but don't worry, because there'll be more next year. But thank you all so very much for coming out tonight to learn more about Pinto Abalone restoration in the Salish Sea with Emily here tonight. Again, she's a shellfish biologist with Washington Department of Fish and Wildlife. And yeah, feel free to go and swap the computers here. She's definitely our people. She emailed me reaching out like, hey, I have this presentation about restoring this important snail in the Salish Sea, and that's all I needed to know to be like, hey, would you like to do a Cocktails and Fishtails talk with us? Because we would love to host you and learn more about this issue. She's somebody who has done a lot on the water, from tall ships to scuba diving, so she's like on the water, under the water, around the water. She's got this place covered, and we are so excited to learn more from her tonight. So, with that, Emily, here you go.
Thank you very much, Stina, and thank you all for coming out tonight. I'm excited to be here. Just getting used to the microphone. I don't really spend a whole lot of time talking into one of these, so let me know if I need to speak up or speak more softly or anything like that. All right, well, enjoy your cocktails. Let me know if you have any questions, and let's get started. So as Stina said, my name's Emily Luce. I'm with the Washington Department of Fish and Wildlife. I'm a subtidal shellfish biologist, and I have a little contents here for you so you know what's coming. I kind of gave my personal introduction. I'm going to introduce you to the rest of the team that I work with. We'll have a little historical overview of abalone, talk about abalone biology in general, a little bit about Pinto abalone more specifically, then we'll discuss the status, our restoration approach, and finally the successes and challenges of restoring a marine snail. First of all, I need to acknowledge my wonderful coworkers. We all make up the Washington Department of Fish and Wildlife Subtidal Shellfish Dive Team. There's currently 8 of us, and we spend a lot of time together. We spend a lot of time on the water and under the water. We do a lot of different things that I'll get to in just a minute. But I also want to acknowledge our very close collaborators with the Puget Sound Restoration Fund. Here on the bottom we have Eileen Bates and Josh Balma, and they are an integral part of our restoration efforts. Oops. A little bit more about the team that I work with. A lot of these people have been part of the team for a lot longer than I have, so I want to acknowledge that. I've been with the team for 5 years now. I started out more in the geoduck side of things and have shifted to abalone because they're pretty special. But we do a lot of other things throughout the year, and we kind of all help out with all of our projects. So you'll see us helping to manage shellfish fisheries around the state, specifically subtidal shellfish fisheries. So anything you need to dive to harvest, we manage. That includes cucumber and then sea urchins for the most part, as well as geoduck, which we just finished. My colleagues just finished the last day of the geoduck field season today, so we're celebrating that. We also have some colleagues in other parts of marine research. We help out with some Pycnopodia projects. We do a lot of collecting for some of the different labs that are working on sea star wasting disease. And we've also done some purple urchin and kelp work out in the strait.
I have a question.
Okay.
When you say subtidal, how deep? You go up, what, 50, 80 feet?
So we typically go to 70 feet plus the tide. The geoduck and some of the echinoderm fisheries can be harvested down to 70 feet plus the tide. So, say the average high tide is an additional 12 feet, then we'll go to 82 feet, and that's about the deepest. For the abalone work, we're super shallow, which means really long dive times in really cold water in February. But we'll talk a little bit more about that in a few minutes. All right, so with that introduction out of the way, let's go ahead and dive in. All right, just a little sneak peek to see what we do. That was one of our 2 research vessels. We have them very aptly named for what we do. We do a lot of counting and a lot of measuring. So our vessel names are the enumerator and the caliper. So if you ever see us out on the water, give us a wave or come on by and say hi. Okay, so now to abalone. This is going to be a very brief historical overview, but the earliest uses of abalone were with Native Americans who actually left middens with piles of abalone shells 12,000 years ago. Abalone hold profound spiritual and ceremonial importance, and they've been used in traditional rituals, artwork, and they also symbolize prosperity, protection, and a deep connection to the ocean. Native Americans were known to eat the meat, as well as using the whole shells as bowls, and they would also use parts and pieces of the shells to form fish hooks and jewelry. You can see in the middle is a woman collecting some abalone from the intertidal area, and then the pictures on each side have some very nice abalone earrings. In the late 18th century, Spanish settlers exchanged abalone shells for otter furs, or even sold them, and eventually kind of traded up for gold. Later, abalone became popular among all levels of society, and this is an interesting day trip for this group. It's they're abalone hunting in the Coronado Islands of Mexico. Now this slide seems a little bit random, but when I was researching historical photographs of abalone, I found that Lee Passmore, the photographer of this photo, apparently had a hobby of putting tarantulas on his face and looking really happy about it. I tried to find the backstory and I couldn't, but it was too interesting and unique to pass up sharing with all of you. Okay, back to abalone. They're especially vulnerable to overharvest due to several of their characteristics. They aggregate during spawning, which we'll get into a little bit more later. They have patchy distribution. They have a short larval period, and they have very slow growth and low recruitment. This combination of things just kind of sets them up to have serious population issues when they're overfished or overharvested, as they were in this photo. Now, I just want to acknowledge a lot of these photos that I'm showing you are not from Washington. I looked around, there's not a lot of historical photographs from Washington that I could find, but if anybody knows of any, I'd love a source. Abalone's popularity as part of the California culture seemed to peak between the 1950s and the 1970s, and this was actually part of a photo shoot that was in Life magazine in 1944. Okay, now we got a little bit of the history out of the way. Let's talk a little bit more about the biology of abalone. So again, I'm going to start real general, and then we'll go in a little bit closer to Pinto Abalone. Pinto are just one of 7 species that are found on the west coast of North America, and they also have the widest range. If you can make out, there's a kind of an orange bracket combined with a yellow bracket. Those are both Pinto Abalone. They were at one point known as a subspecies, the threaded abalone, but it's all the same. So you can see that they have a much wider range than the other abalone. So, what is an abalone? This seems like a really basic question, and you're all here, I'm pretty sure you know what an abalone is, but I just thought it might be fun to dissect this a little bit. So it's a marine gastropod. But what does that mean? Did anybody have Latin in school? Yeah. So then you know that a gastropod basically means stomach and foot. So if you look at abalone anatomy, this pretty much sums up what they are. They're a big stomach and a big foot. Gastropods are also a type of mollusk, which I'm pretty sure you're all familiar with as well. But let's just look at that a little bit. So we ask, what is a mollusk? Mollusks are highly diverse in size and structure, but also in behavior and habitat. They can be freshwater, terrestrial, and even marine. So a mollusk is a soft-bodied invertebrate, often wholly or partially enclosed in a calcium carbonate shell. It's one of the most diverse groups in the animal kingdom, and there are nearly 100,000 described species. So just to solidify this in your mind, we know now that abalone are mollusks, but also all of these clams, bivalves, oysters, a lot of the shellfish that you may eat around Puget Sound are mollusks, but so are these. These are the nudibranchs, known as sea slugs or sea snails. And also octopus, cuttlefish, and squid are mollusks. And these are actually among the most neurologically advanced of all the invertebrates. So there's some good trivia for you. And we haven't even covered terrestrial snails and slugs, but we're gonna skip that part for tonight. Okay, some more biology about abalone. They do have some very distinguishing characteristics beyond their unique shell. They have what's called a radula, which is a rasping tongue with chitinous teeth, and they use these to clean diatoms and microalgae off of rocks on the reef. They also have a broad muscular foot, just as we talked about with the gastropods, they are made up of a big foot. And this foot helps them cling to rocks, and then they have musculature that can pull the shells down over them, so that helps them defend against their predators who want to come up on the rock and just snatch them off. They're actually incredibly strong. And they also have these respiratory pores that you can see in the photo on the right. They're kind of yellowish in this photo, and these have kind of multi-purpose use. They are used to vent water from the gills, to discharge metabolic waste, as well as releasing sperm and eggs for reproduction. Another kind of famous thing about abalone is the nacre, or the mother of pearl on the inside of their shells. And the nacre is actually composed of microscopic calcium carbonate tiles, or aragonite tiles, that are stacked like bricks and held together by a biopolymer or a clingy protein substance. This picture on the left side is actually from a scanning electron microscope, just to show you what the actual nacre looks like in super zoomed in lens. And you can see it's kind of stacked like tiles. It makes a really pretty pattern. These substances are actually being studied by material scientists from around the world for many different uses. I have a few here. Some of the more interesting ones to me were that it's being used in the medical field. They have a nacre powder that they put in bones to help the bones form around them, and are also used in implants. They're used in a lot of different engineering and transportation needs. And they're even used in studying ancient climates because the nacre actually encodes environmental info within the cells. And now reproduction. Abalone are broadcast spawners, which is fairly common among the other snails and sea creatures in the mollusk and gastropod family. So they release sperm and egg into the water. And because that's how they reproduce, they really have to be in close proximity for success. So this is really unfortunate when you have a population that's starting to collapse. I wonder how many people in this room are seeing the abalone in this photo. Does anybody see them?
Two.
Yeah, well done. Got a room full of abalone sighters. There's actually two, and I just circled them here in case you're having trouble seeing them. They do a really good job of camouflaging with the reef, and it's super special to find them, but it's really special when you find two in such close proximity. You just hope they hang around that close until it's spawning time. Okay, and now we're going to zoom in a little bit more to the Pinto abalone. Heliotis kamchatkana, which in Greek, heliotis means sea and ear, so they kind of resemble an ear with their spiral. As I said earlier, pintos have the largest range of any of the abalone on the west coast of the US, from southeast Alaska down to Baja, Mexico. They are generally at less than 40 feet of depth, and they're known to be a little bit shallower in the northern part of their range and a little bit deeper in the southern part of their range. They're also the only species of abalone found in Washington, so that alone makes them pretty special. They are found in and around the San Juan Islands and also in the Strait. And you can, it may be hard to read that text, but the hatching that goes down the coast is actually otter habitat. So you can see inside the western part of the Strait, there is some overlap with the abalone and otters.
Which one's the abalone on
It is kind of the diagonal lines that go from the northern end of the San Juan Islands out to the western extent of the strait by Nia Bay. In terms of their specific habitat, they prefer rocky reefs, and they're almost always associated with kelp beds. Sometimes they're a little bit farther from the kelp, but generally in close enough proximity to get some good food. And they play a very vital ecosystem role. They have actually earned the nickname Roombas of the sea because they do such a thorough job of kind of vacuuming and sweeping and cleaning up. They like to settle on rocks, like this one is here, and clean off kind of this film of diatoms or macroalgae. They also play a really critical role in nutrient cycling since they do take up those nutrients from the macroalgae, and they clean spaces on rocks so that a diversity of kelp species can settle and establish on the reef. Okay, so now we're going to skip ahead to a timeline. I want you to keep in mind that this is a super broad overview, so we're going to go through the timeline of Pinto Abalone, and then we're going to step back a little bit to talk about the work that we're doing. So I like to call this the timeline of Pinto Abalone status in Washington, or how the Pinto Abalone went from harvested species to endangered species in 60 years or less. It was a pretty rapid decline, and we'll see about that in a minute. I also want to mention this artwork in the lower corner is made by a really talented woman named Andrea Dingledine. She goes by the local naturalist. And I have some stickers with her art for afterward if you want to come up and grab one. Okay, so let's start out in 1959. This is when the recreational fishery was authorized in Washington, and the personal harvest limit was 3 abalone per person per day. So keep in mind this is sport harvest only. There was never a commercial fishery for abalone in Washington state. In 1980, they started to see declines, and so they increased the minimum harvest size to 3.5 inches. 1990, further increase up to 4 inches now, and they also introduced gear restrictions and a reduced daily limit. Now I want to pause here for just a minute because the 1980s and 1990s were known for rampant poaching of abalone in Washington state. The picture on the bottom is actually a truck from British Columbia, but it was found full of Pinto abalone that were all illegally harvested. And the boat in the upper, toward the upper right, was actually purchased by someone who made his fortune poaching abalone. I don't know if any of you are familiar with the Seattle Times reporter, Craig Welch, but he wrote a book called Shell Games. See some nodding. It's a pretty good book. If you haven't read it, you might want to pick it up. But he tells the story of the owner of this boat who was rampantly poaching abalone and basically got caught. But when he got caught, he just told the cops everything, to the point where he became an informant for the Department of Fish and Wildlife, and he let them know about all the other poaching rings that were going on in the state. Unfortunately, in 1994, there was an explosion on his boat. He decided the other poachers had found out, his cover was blown, he thought they were all kind of retaliating against him, so he resigned and left town. Apparently hasn't been seen since.
Was the poaching for the meat or was it for the shell?
It was primarily for the meat, as I understand it, but I think the shell probably had its own value. Okay, back to our timeline. So now we're in 1994 and the recreational fishery has closed. They've decided the declines are significant enough, and I'll show you a graph in a minute that kind of backs this up.
Can you explain the size of the circles and the colors? Does that mean anything?
Oh, not really, it's just the design. And I did, like, as I said, it's kind of a broad overview. It's kind of the highlights and the lowlights.
Yeah.
There's a lot of detail we're missing.
Thank you.
But yeah, absolutely.
Look at me like at 20.
Okay, so now we're in 2003, and this is the point where our agency, the Department of Fish and Wildlife, as well as University of Washington and the Puget Sound Restoration Fund, in coordination with NOAA, launched a restoration effort. By 2017, the Department of Fish and Wildlife had documented a 97% decline in populations at index sites that they were monitoring in the San Juan Islands. And again, we'll skip back and find out more detail about this in just a minute. Finally, in 2019, our agency formally listed Pinto as state endangered, which is the title that it still maintains today. So what's after that? Well, the present and future, but we'll get to that in a minute. Okay, so as I mentioned, we're going to back up a little bit to explain part of the timeline and how we got to where we are. In 1992, the Department of Fish and Wildlife established 10 index sites around the San Juan Islands. And the goal of the index sites was to find known aggregations of abalone, or basically an aggregation is any time there's more than one within a limited area. So divers from the Department of Fish and Wildlife found aggregations around San Juan Islands and established sites there that they could go back and basically reproduce the counts that they found. So they started out by going back every 2 years to check up on these known aggregations and see how their numbers fared over the years. Okay, here's the graph that I mentioned we were going to get to. Now, all the information that's kind of smaller isn't super important. On the left side is the density of abalone per square meter, and across the bottom are the years that the surveys were done. Again, the detail isn't important, but what you will immediately notice is the sharp decline. And you also see the dotted line is the presumed minimum density for fertilization. This was kind of based off a number of things, including collapse of a different species in Australia, some modeling of white abalone populations, and lab work on other marine invertebrates. And in 1994, you see the fishery closure, but you also see that, unfortunately, the fishery closed after the populations dropped below the minimum presumed density for successful reproduction. Okay, and now just to pull out a few numbers, in 1992, within, again, these 10 sites in the San Juan Islands, 359 abalone were recorded. By 2003, that number had less than halved to 136. And finally, in 2017, there were only 12 abalone left on those sites. So if you do the math, that's a 97% decline in just 25 years. It's pretty startling, and something had to be done.
What was the cause of that decline?
It was many different things, some things that we probably can't even quite get at, but over-harvesting was a big issue, illegal harvesting and poaching. But there's also potential environmental issues that are kind of hard to tease out. But the biggest problem is they suffered kind of all these detriments to them, and then there were too few of them to be in close enough proximity to reproduce.
How long does an abalone live?
They live at least 15 to 20 years, possibly up to 40. As far as I know, no one has figured out how to age them yet. Yeah.
How does that decline relate to the abalones in California, in the Southern California coast, the pinks and the reds and the greens?
Yeah, I don't know specific details about that. I could definitely get that information and get back to you.
If they're linked.
Yeah, I'm not sure. I know that they had some of the same issues, over harvesting and
You
poaching.
can't buy it in a restaurant anymore, or
Right.
in a seafood place.
Right, yeah.
Are abalone only native to like the western coast of U.S. and everything, or are they distributed worldwide?
There's actually a distribution worldwide. There are none on the east coast of the U.S., but there are some in Australia and other parts worldwide. I should have brought a map of all of the different species.
Are there abalone, were there abalone in Puget Sound ever?
Not that I am aware of. There have been some documented intertidally, even up to recent times, but I'm not sure how far into Puget Sound they historically reached. I've never really seen anything documented where they were in the sound. And that may just be a combination of different water quality characteristics and salinity and things like that.
It's been trade items.
Okay.
There must have been trade items back.
So we just kind of went over how we had this precipitous decline. And the other thing to note about this same period of time was that the abalone shell length continued to increase. Now this might initially sound like a good thing. If the average size of the shell increases, it means they're getting older, they're staying alive, basically. But in reality, it means we're not having any new recruits to the population. So there are no small abalone to bring that average down, which basically means we have an aging population. So you can see there's just kind of some of the highlights off to the left. 1979, they were just under 100 millimeters on average, and by 2017, they were 127. So there are other factors, as I kind of briefly mentioned, that can't be ruled out. Disease, climate change, over-harvesting, poaching, but reproductive collapse due to the low adult densities is most likely the explanation.
So is there any attempt to increase the population by, farming them basically?
That's what we're doing. I'm
Okay.
gonna tell you all about it.
Does the water temperature or salinity have any effect on them?
It's possible that it does. I'm gonna talk a little bit about that later. We have looked at that, but haven't really teased anything out of note. And by we, I should say our collaborator, Eileen Bates, just finished her PhD at the University of Washington. We'll talk about her in a bit, but she has been working with us over the last several years, and she looked into all of that, and there's no smoking gun, so to speak.
Do they procreate throughout their lifetime, or are they like humans?
Yeah, they're like humans. They definitely need, we typically think of once they reach 50 millimeters in size, they are kind of reproductively mature. But they also have very variable growth rates, so it's hard to say at what age or how many years old they would be. And then surely, like most animals, at some point they probably age out of reproduction, but I'm not sure at what point that would be. I think it might be very individual. Okay, so once again, we're gonna back up a little bit and talk about letting the Pinto Abalone restoration begin. As I noted, a number of agencies have seen this decline over a period of decades. And at this point, the Department of Fish and Wildlife, the Puget Sound Restoration Fund, and other collaborators have decided that we need to think about captive rearing methods to help the populations. And the goal was always to create a self-sustaining network of reproductive populations in Washington. Okay, so the first step in the recovery program is to collect abalone in the wild, because in order to make little abalone in the hatchery, we need some brood stock. So I'm going to play this little video that shows you how we collect abalone. Fun fact about abalone that I didn't mention yet is they're actually hemophiliacs. So if they get a cut or a puncture, they have no ability to clot their blood, so it's really dangerous. So when we do collect them in the wild, we have a technique of using a sea star. This is my coworker Katie. She has an ochre star or pyzaster, and she's going to hold it on that abalone there. Once the abalone kind of senses the sea star, which is one of their predators, it decides it doesn't want to hang around on that rock anymore, and it starts making a run for it. So as soon as Katie sees that, she just gently kind of bumps it off the rock, and there we go. So that's an abalone. I think I forgot to mention that they got their common name Pinto because they have this mottled flesh that looks kind of like pinto coloring or a pinto bean. All right, and if you don't believe me that that abalone moved because of the sea star, I have more evidence. This is a Pycnopodia, which is one of their main predators.
Drama. Run!
Which is the fastest?
Is it a sprint or a long haul?
What sense do they use to know that the starfish is there?
I just wanted to leave the audio on and didn't want to talk over it. But no worries. I believe they sense pheromones in the water. They can tell that they're coming. They have kind of eyes, but they're not very advanced. They wouldn't be able to see them approaching. So that's what I like to think of as a high-speed chase. Okay, so once we have the brood stock, we take them back to the center that the Puget Sound Restoration Fund runs. It's the Kenneth Chu Center for Shellfish Research, and it's part of the NOAA research station in Manchester, Washington. And you can see they have a good grow-out facility. They have nursery buildings. They can produce microalgae for feeding the kelp, for feeding the abalone, as well as kelp. And they also have some additional ocean acidification research going on there.
Can you visit there?
I don't think that it's open to the public. I know during COVID it was very, very locked down. It was hard for us to even get in. But I don't know if they do, I'm not sure if they do tours or if something could be arranged. It would be worth looking into. Minor technical difficulties. Oh, I bumped it with my leg. Thank you. Okay.
Stina, set this tour up.
Oh,
Yeah.
oh,
Mhm.
okay.
I just agreed with
I'll see
you.
what I can do.
I can get some connections if you don't already have them.
Great.
Okay, so at the Chew Center, the Puget Sound Restoration Fund does all the hatchery management, and specifically our close colleague Josh Bauma is really responsible for basically the whole Pinto Abalone program there. He has developed innovative culturing techniques over a series of years, and he always ensures that there are many different crosses. So we'll have individual female and males that we collect from the wild broodstock, and he'll make as many genetic crosses as he can to ensure genetic diversity when we do our outplants. We also use only first generation, so there will never be outplants that were spawned from other abalone raised in the hatchery. It's always just from the fresh wild broodstock. So in 2009, the first outplants went out. We'll get into a little detail about this in just a minute. But basically, since then, Josh and PSRF have produced approximately 55,000 juvenile abalone that we have outplanted. And he has kept track of 250 genetically distinct families. Also want to mention additional collaborators. Recently, the Seattle Aquarium has gotten their own hatchery going. It's pretty exciting. They are now doing everything from the culturing of the abalone to the outplanting on the outplant site. So we've got a really great relationship with them and really appreciate their work. And also the Port Townsend Marine Science Center is kind of functioning as a grow-out facility. They aren't culturing their own, but when PSRF's tanks kind of get too full, then they'll send some out there and they will keep them alive for us and let them get a little bit bigger and stronger. So once the juveniles are raised to a point where they're large enough to go out in the wild, Josh and his crew will very carefully and strategically place these tiny little abalone into these PVC tubes. And they're always very careful to ensure a mix of families within each tube, and each site typically gets 3 to 4 tubes. So there's a good mix of genetic families going into each site. Next, we'll take those tubes and we'll put them on the boat. We collect fresh seawater every morning for them to hang out in until we get to our sites. Oh, and one other thing I almost forgot to mention. If you see the green tags on the baby abalone on the left, sometimes we have 2 different cohorts. There will be one cohort that's approximately a year old or has been raised in the hatchery for close to a year, and then a second cohort that's about 2 years old. So they typically mark the second years just so that when we see them on the sites, we know how old they were and we can kind of see what their growth has been over time. So the next part is actually the outplant sites. Now by the time we get the juveniles out in the boat, ready to go, we already have the outplant sites set up and established. But basically, what we do is set up the outplant sites in a way that they'll appeal to abalone, or at least we hope they will. Clearly, as humans, it's pretty hard to imagine exactly what a marine snail would like, but we do our best. So we always look for good habitat. We look for a complex reef structure that has a lot of refugia for pintos to hide in, which is pretty easy when you're talking about a 5 or 10 or 15 millimeter snail. And then we always look for kelp, either nearby or sometimes actually on the site itself. And this kind of scratched little drawing we have, this is what we actually use to find our sites year after year. So we have GPS coordinates to get the boat there, but that only gets you so close, it's not exact. And then we'll look for the characteristics on the specific map for the site. It's not always as easy as it seems because there's always kind of like, all the boulders look alike after a while. Some of the maps will say like table-sized boulder, and you get down there and there's like 18 table-sized boulders. But it's always a fun challenge to find them. We usually do a pretty good job. So that is the map. And then once we have our baby abalones, we go ahead and hop in the water. On the right here is Katie. And you can see in her yellow mesh bag that she has some of those PVC outplant tubes and is hopping in the water to deliver them. Okay, so this is one of our outplant sites. There's not much kelp on this one. I know I said that's a feature of most of our sites, but it just makes it easier to show you a good photograph of the overview of the site without kelp in the way. This is actually our site called land crab, and you might pick this up in a minute, but basically all of our sites are named for World War II amphibious assaults. And I looked this up because I don't know all the history of all of our sites, but land crab was the Allied amphibious landing on Attu Island in the Aleutians in 1943. So I think it's very interesting. Clearly we need code names for these spots because we don't want to publicize to the general public where we're putting, 800 endangered marine snails every year. But if you want the details about why they're amphibious World War II assaults, you'll have to ask my boss. He came up with that scheme. Yep.
So the abalone crawl out of the PVC tubing to get on the rocks and everything. Can you go back and reuse get the tubing back and reuse it?
We do, yeah. You can see that they're kind of stuck in little nooks and crannies, and they have cobble and boulders piled on top. And part of that is just so the baby abalone don't get thrown around in the current or the swell surge when they're down there, but also because we do go back and we collect all of those for reuse. We certainly don't want to leave plastic out on the reef. Okay. So again, as I showed you in the drawing of this site, we basically have good habitat. There's a ton of boulders there that you could imagine little tiny Pinto Abalone could get kind of crawl up underneath and in the cracks and find lots of good places to hide from predators. And there's also a lot of this Christos coralline algae, the pink stuff that's all over the rocks. That seems to be a very good indicator of where pintos are going to find good habitat. And just to show you how cute little baby abalone can be, this is what they look like within the tube. And you can see they've got their epipodial tentacles out, they're kind of sensing their environment, and they're thinking, I want to get out of this plastic and onto that rock. And then we pan out, you can see another tube in the background, some rocks, and there's some pretty good current going on that one. It seems to be another characteristic that abalone like. But again, it's really hard to tell.
How long do they stay in there since they're on the move? I mean, you go back in a week and they're empty?
We don't typically go back that quickly, but when we do go back, they're usually all gone. I think it really varies. I think some probably climb out almost immediately, and some probably hang out for a week or two. It would be interesting to go back really soon and see, also see if it's associated with the size.
What's the survival rate?
We're going to get to that in just a minute. Okay, so we put our juvenile abalone out there. Now we come back, we want to see how they're doing. We want to see how many are still there. So what we do is we go down underwater, we re-find our site, and then we basically string out lines. We do the perimeter. We have they're called raft dogs. They're little metal spikes that we hammer into the rock, and that's how we delineate the corners. So we'll find the corners, run a line, and then we'll actually run lead lines to divide each plot into lanes. And that's purely so that we can make sure that we cover every square inch. So once we have it all set up, we'll send divers down to actually do the very methodical kind of, I like to say crawling over the reef. Obviously we don't touch it, but we go very, very slowly and methodically. It generally takes, these sites are roughly 10 by 10 meters, and it takes at least 2 to 3 hours to survey an entire site because we're just moving that slowly. We also, in addition to doing the lanes, we also do a perimeter. So I've kind of shaded the area that we do. We generally do 2 meters on each side of the perimeter of the plot. And that's just because we want to give them a little bit of allowance, just because we have these delineated lines of where the plot is. abalone don't really pay attention to our conventions, so we want to make sure if they've gone just off the plot that we're still going to see them and count them. So I have quick videos so that you can see a little bit about what it's like down there. You can see there's a ton of habitat. This site has kind of more of a bedrock sediment, not so much boulders, but there's still plenty of habitat. Looking around at the divers, you can see the divers have a slate, and they're basically, whenever they find a juvenile pinto or even an adult, sometimes there are adults on the sites, we will measure them, we'll record if they have a tag or not, and then we'll also record in what lane they were found. And also if we find a mortality, a shell without an animal in it, then we'll record that as well. So you can see the divers are just kind of hanging out. They're not really going anywhere because there are so many little nooks and crannies to peek in. You can see some of our lines set up there too. Okay, so now we've done all the fun part. Let's talk about the survival and see if we're actually having any success here. So this graph shows survival of outplants after 1 year. And these are what we consider the 2-year cohort. They're basically 20 months old, but we just consider them 2 years. So there's a few different things I want to bring to your attention here. First of all, each bar in the graph is a different site. Again, the names are across the bottom and they're not important, but you might notice that they are the amphibious assault sites from World War II. Other things to note, for the first 8 years of outplanting, we actually tagged the abalones with unique ID numbers so that we could go back in subsequent years and then see if we found additional abalone that we didn't see the first surveys. So that's what those orange bars are on top of the blue bars. The sites were outplanted in different years, and many of the sites have been overseeded in subsequent years because we like to go back to a site once we have success there. And an example of this is Gold, which is the third from the left. That one, we just put abalone on, more juvenile outplants in April of this year, but this was all from the original outplanting. And finally, in 2019, we started the mixed age cohorts. So we outplanted 1-year-olds and 2-year-olds, but the graph for 2019 only represents the 2-year-olds. Also, the blue line you see is what we consider a good site or a successful site. In this case, it's about 4% survival. And now, if you look at the first chunk actually up until 2016, we had 0 to 23% survival. So that averaged about 10% compared to the second bracket of sites, which averaged 4%. The difference could be partially explained because the second set did not have repeat survey data. So they were only surveyed once and counted once. So it looks a little bit less. But if we had gone back, we might have had closer to 10% survival there as well.
Do you know the natural survival rate?
We don't. It's really hard to find. And in this case, we put so many out there on the reef and we get such a small percentage of survival. But at the same time, in the hatchery, there's a lot of natural mortality as well. There are lower trophic level species, so that's just kind of how things work. They reproduce and produce a whole lot of babies, and then very few survive.
You presented a rapid decline over time of the species, and you also indicated that there were multiple reasons and it's difficult to tease out the data. And we see the the high mortality rate here. And so I'm wondering if the state would be interested in, or if they are, conducting research parallel, because my concern here is this almost seems like rearranging the deck chairs on the Titanic. If you have a 4% survival rate, you got to address the bigger issue in the room, and that is why the species has been declining. So have efforts been put into that as well?
Yeah, we have. I'll talk about that a little bit more later. But again, part of the 4% survival is it's just the nature of a lower trophic level species. So you're just gonna have a whole lot of babies being produced, but then you're gonna have a really low survival rate in the wild, in the hatchery, in any case. So to us, 4% seems acceptable, even good. And I know it sounds really low, but it's just kind of the nature of it. I would say most sites we outplant between 700 and 800 juveniles. So if you get 4% of that, it's still more than enough to have a breeding aggregation, and that's what's really important. It's just making sure there are enough individuals there in close proximity that they can be successful with their reproduction.
Are they dying because there's lack of the right environment, the right food, or are they being eaten?
Yeah, those are all really good questions that I'm going to get to in just a minute, and I don't have super satisfying answers, but we'll talk about it. Okay, so I showed you the 2-year survival. Now this is the 1-year survival after the first year of being outplanted. Sorry, I'm not usually used to talking this much. Okay, so in this slide, again, the sites are in order of establishment. So the sites that were established first are to the left, and so on. Oops. Sorry about that. So we did start placing 1-year-olds out in the wild on the sites until 2019. It had always been 2-year-olds before then. So this is looking from 2019 up to 2023. And what we saw was that after modest survival of the younger abalone in 2019, we went back in subsequent years and started putting them on newer sites. So again, you can see this blue dotted line is what we consider a good site. And for the 1-year-olds, it's only 1%. Because again, you have more, they're smaller, there's any number of things that can happen to them. But we felt like if we get 1% survival, that's pretty successful. And again, when you consider having abalone in the hatchery, there's a lot of added costs and work and natural mortality that happens between 1 year and 2 year. So the hatchery folks are kind of trying to sort out the balance of, is it better to only raise them for a year and then get them out in the wild, or raise them for longer? It's kind of a cost-benefit analysis.
So may I ask you a question?
Yeah.
So your grid that you showed about where you count, I'm wondering about how it affects the survival rate, because clearly some of them leave that area.
Definitely.
Correct? And maybe some of them are in the area, but they're all down in the rocks and you can't find the little buggers anyway.
Yeah, these are all things we've acknowledged but don't have a great solution
So
for.
I'm guessing that this algebraic equation has a lot of factors in there that
Yeah.
eventually come out with 1% at the end.
Pretty much, yeah, that sums it up.
Thank you.
Yeah, of course. Okay, so again, 1% seems lower than the 4% we saw with the 2-year-olds, but it's a lot easier for a larger animal to survive longer in terms of predation and assorted variables. And if you take into account the added time in the hatchery from 1-year-olds to 2-year-olds, there's a lot of natural mortality that would happen in a hatchery that isn't happening there. So it's naturally going to happen out on the sites after they're outplanted. Okay, now we're going to get to what a lot of you have been asking about. I just showed you graphs where roughly 1 to 20% of the abalone were there after the 2nd year. So that brings up what happens to the other 80 to 99% of the abalone. That seems like a whole lot to not be able to account for. So there's a few things that could potentially explain this. One is mortality. We kind of talked about this a little bit. We do recover empty shells. As I mentioned, we measure them and note that we found them. But even the empty shells only account for 1 to 5% of the numbers from the outplant that weren't accounted for by live animals. Were they washed away by current or broken? It's very possible. Some of these sites have really strong currents. A 5mm abalone shell weighs next to nothing, so they may just get blown away and we never see them again. Are the shells consumed by predators? Again, very likely. Several years ago, our group did a time-lapse camera study, and I think we had a total of over 100,000 images. We set the cameras down there and they recorded every few minutes. But after looking through all those images, there was nothing obvious. There was no octopus just chomping all of them. There's no big fish, kelp greenlings swimming by and grabbing them. So we really don't know. There was Western Washington University Shannon Point, RU students did a project this summer and found that some snails are responsible for abalone predation at the size where we outplant them. But we don't have all the details on that, so we're looking forward to more details there, and hopefully we'll plan more studies about predation in the future. Another possibility that you also brought up is that they moved away. So as I mentioned, we do survey the perimeter in hopes to kind of grab those that might be moving off site, and by grab, I don't mean we're not going to place them back on the site, I just mean to count them. And we have actually documented when an entire cohort moved. We have a site called Juneau that we went back to and counted, and the numbers were so low that we decided it was not a successful site, and we stopped outplanting there. And then later our divers went back to recover a piece of equipment and found that there were a whole aggregation about 10 meters to the west of our original site. So we ended up actually extending our site and then starting it all over again, and it's been very successful since. And finally, kind of the third option is that they're just too hard to see. As I mentioned, there's tiny, you saw the photos, there's a ton of nooks and crannies. So where are they going? We don't know. It's really hard to count how many we're not seeing. The other thing that is interesting is that repeat surveys show that our detection rate is low. And this is obvious. It's really hard to find stuff that's up under and cracks. And I mean, we're like doing handstands and contorting ourselves to see everything that we can. But it's inevitable that we're going to miss them. And we've seen in repeat surveys that our detection rate is between 20 to 40%. Which is actually good news. It means there's a whole lot of abalone out there that we don't even know about, which kind of increases our success rate. This is the other big question we have. What makes this site successful? Again, it's really hard to know because we have human eyes. We don't have abalone eyes, snail eyes. So we do the best we can. Observations on all of our sites suggest that food is not limiting. There's very frequently kelp on the site, as I said, if not, it's nearby. Is there direct predation? We kind of covered this. We didn't see anything in our camera surveys, but it's possible there are things going on that we haven't documented. And then this is the last thing that I mentioned a little bit earlier, is that it could be oceanographic conditions. And the newly minted Dr. Eileen Bates, Eileen just defended her dissertation a few months ago at the University of Washington. And she worked with us for years to put out these sensor arrays. This is actually Eileen in the photo. And you can see her sensors are kind of cobbled together on 2 blocks. But she was looking, and you can actually see the tall thing sticking up as a current meter. So she was looking at pH, dissolved oxygen, temperature, salinity, current flow, and light penetration. And she had gobs of data. I don't even know how to express how much data she had. We did this for years, and it was kind of constantly collecting data, and she found nothing that clearly stood out to her. So we're guessing that there is some kind of oceanographic condition that affects this, but we don't have a direct answer. And I think more work will be forthcoming on that. Okay, so now we're going to go back to the index station graph again, but this time I've included this year's data. We surveyed these in 2024, and you might notice a big jump between 2017 and 2024, but we had a little issue called COVID. So we kind of lost some field work. We had a longer span of time than normal between these 2 surveys. And just when we were getting all of our field work together again after COVID, we had an engine blow up and then we were down a boat. So we had some time when we didn't quite get back to these index sites as quickly as we wanted to. But the good news is if you look really, really closely, you can see a very slight uptick. So,
We'll take it.
this is so small, but also it's a victory. So,
Yay, snails!
this was the first time in 36 years of monitoring these index stations that we had an increase. Unfortunately, yeah.
Really evident.
That must be it. That's it. They wanted a break. They're like, retire these human eyes. I'll take that back to the team and see what they think about it. But yeah, so I don't want want to get you really excited and then let you down, but we only found one more than we found in 2017. But one is one, it's more is more. And also, we've already talked about how we're probably missing a whole bunch, so hopefully there's more out there. In any case, sometimes small successes can feel very big. We're very excited about this. The other thing to keep in mind is that in 2024, we also did perimeter surveys. I showed you that we normally do those on the outplant sites, but in the past, we haven't done those on the index sites. So because we hadn't had that data before, we had nothing to compare it to. So we didn't count that in this graph where we're saying we found one more. But we actually found 23 abalone in the perimeter surveys. So how many were there before? Hard to tell, but there are more out there than the 13 we documented. The other good news is that we also documented a decrease in shell length. And again, this seems kind of inversely positive, but if the average length of shell decreased, then we're having smaller abalone that are recruiting. So that's a really good thing too. So now you might ask, where do we go from here? There's a lot of things that we can do in the future. We have a lot of ideas for research and plans. But first, we're just going to continue developing new outplant sites and overseeing the established sites. We have our way of determining what we think was a good site, so we're going to keep overseeding and hope that they continue to be good sites. We're also considering expanding into the Strait of Juan de Fuca. We've done a little bit of field work out that way. We have done genetic testing of the abalone in the strait and have found that they are not genetically different from the ones in the San Juan Islands, so there shouldn't be an issue there. And we're going to do some kind of preliminary surveys in the next month or two to see about the populations out there. We've been kind of monitoring them in general over the last 4 years during some of our other work. We have a lot of urchin and sea cucumber surveys that we do out there. We always note abalone when we see them, but this year we're going to have a more targeted approach to surveying. We're also going to continue monitoring our previously established index sites, the ones that you saw the graph of a few times, and we're also establishing new sites, which is pretty exciting. We started some new index sites this year that we're looking forward to going back to in a couple of years to see if they are maintaining their abalone as well. And finally, we are hoping to establish more collaborations and partnerships. We've really kind of branched out over the past few years, having more boats running. We currently have 2 boats that are up and running. Hopefully they stay up and running. And we've actually had some weeks where we have 2 teams. Basically, our whole group will go out on 2 different boats and we can get a lot more work done. And with more people helping us culture and raise abalone, we have more to outplant. So it's all good news. Finally, I want to, again, acknowledge and thank my teammates. Several of my teammates have been doing this a lot longer than I have for the agency and work in abalone in general. So I want to recognize that, as well as our colleagues at PSRF. I also want to thank Stina and the whole Harbor Wild Watch team, and all of you for coming out tonight. Thanks so much.
Can I ask, what are your funding sources for all this research?
A lot of it is state funding. So we go to the legislature every year, not me personally, but we work on funding from the state, and we got a biodiversity package within the last biennium that has allowed us to hire more abalone biologists and focus more of our fieldwork effort on abalone. We don't, working for the state, we don't get private funds really, but we are able to provide pass-through funds to organizations like PSRF, as well as tribal entities that want to help out with our research.
If you increase the population of these Pinto Abalone, how does that increase other species or other interdependencies of the underwater species?
Yeah, that's a good question. They are a really important species, as I mentioned, just for cleaning the rocks and providing area for kelp to settle. And it's kind of the whole kelp forest ecosystem is, a really tightly knit ecosystem where everything benefits from each other. So they're cleaning off more space for other species to settle on, more kelp to grow. And then the kelp is its own kind of ecosystem. So more kelp is beneficial for any number of things. Fish like to hide in the kelp. There's a lot of nursery habitat for fish and assorted invertebrates that could benefit from having more kelp.
Is somebody trying to do the same study on how you make more kelp?
They are actually.
Yeah.
That is definitely in the works. Yes.
Has there been any, like talk about leveraging current, like robotic cameras underwater to do the, like survey and everything instead of having divers down there all the time. And then with AI, maybe have some kind of like way of detecting them or.
It's possible. We haven't really explored that angle yet. I personally don't want it to happen because I want to keep diving as much as I do. It's by far the best part of my job. So I'm not looking to be replaced, but it's possible. At this point, and robotics is by far not my specialty, but at this point it's hard to imagine how you would train a robot to search for something in such a complex environment.
Or even taking the footage data and just having a human eye kind of, look at it to start with.
Yeah, it's entirely possible, but again, let's not even talk about that because I'm going to keep diving.
What I was saying about not disturbing those guys, I wonder, what the stuff that you're wearing and all that air that you're putting out there, I wonder if that doesn't disturb them. Have you ever thought about the neoprene or whatever? Can they sense that change? Or, they're pretty sensitive.
They need to see.
I can't really say. Who knows what they're sensing in terms of that. I know that they don't really behave differently when we're around. That's in part why I showed you that second video where the picno was kind of causing that high-speed chase. Because the first video was easy to say, oh, well, the diver's hand is there, or the diver's right there, maybe it's human. But I will say that without a sea star, we don't have a chance of getting abalone off a rock because they just don't want to go unless they are in danger for their lives, at least from what I've seen. So who knows? I mean, surely there's, neoprene, microscopic particles. But yeah, I don't know. Christina?
Very important question. I see your team has a lot of little creatures in their photos. Does Ocean have a chicken?
Ocean does have a chicken.
Amazing.
Confirmed.
I was hoping it was like a pigeon guillemot.
Great. I love it. I love it.
Do you mark or tag any of your abalone?
We have only marked the juveniles for outplanting, the ones that come from the hatchery, and only when they're the second year. And that's just to kind of differentiate between the 1 and the 2-year-olds. We haven't ever done any tagging of, well, that's not true. When we collect wild abalone for broodstock, then we tag them so that the hatchery can keep track of who's who for, genetic purposes when breeding. But we've never tagged wild abalone out on the reefs.
Are you able to follow any of those markings? I'm not sure what you use from year to year to year.
Markings in terms of the tag numbers?
Yes.
We did. There were select years where they were tagged individually with numbers, the juveniles that were outplanted, but that hasn't happened for years. Typically now it's just a dot. In the picture I showed it was a green dot, but sometimes it's a blue dot or a pink dot. So we don't track them individually. It's just incredibly time consuming, and I've never had to do it for the juveniles, but it's incredibly time consuming on the hatchery staff to put tiny little dots on thousands and thousands of tiny little abalone. So it's something that we haven't really done a whole lot of, but maybe in the future if we feel like it would be beneficial again.
Yeah, I think it'd be really interesting if you could follow, a blue dot from one year to the next, to the next, to the next, even if it's a group.
Number 7.
And look at the growth, between
We do that. I mean, we do. When we see the dots out on the reef, we'll make a note of what the color is, if they have a tag or not.
Okay.
And if they do, what color it is. And then we can kind of extrapolate, average size and growth, but it's just, we're not down to the individual level. It's just too many thousands of baby abalone. Yep.
So you guys are working on the supply side of the abalone, correct? Trying to increase. What's happening on the demand side? Because if world population is increasing, then I gotta believe there's great pressure for food.
Yeah, I mean, I want to say, in terms of Pinto Abalone, I hope nothing is happening, but I'm also not that naive. So I know there's still poaching going on. It's one of those things that's really hard to track. I'm certainly not enforcement. We have some really great people that work for Fish and Wildlife Enforcement, and we spend a lot of time with them. They take us out on their boats. But trying to find abalone poachers is kind of a whole different level. And I'm not aware of any poaching operations that are as big as some of the ones that have been busted in the past. But yeah, it's like the baby abalone. It's hard to count what you don't see. It's hard to know what's happening that we don't see.
Thank you.
Yeah.
Awesome.
All right, thank you all. It's been a pleasure.
I've been sitting here all night trying to think, what is my snail pun for Emily tonight? I would say, let's give her a round of applause because she snailed it.
Nailed it.
Snail class.
Oh, thank you. My heart. So thank you all for tuning in. Thank you all for joining us tonight, for your great questions and your enthusiasm to learn about this important marine species. I think we can all leave tonight knowing a lot more about the abalone and hopefully being inspired to take some action to keep learning about the things that cause us wonder and taking action to protect this incredible place. So thank you, thank you, thank you. Learn and have fun.